All right, now this first like 5 minutes of this little discussion today is going to be review. After that, we're going to get into the sort of the nuts and bolts of mitosis and the different stages involved. But first of all, when a cell is not dividing, it's said to be in what stage of the cell cycle? Interphase. And remember, inter means between, so it's between cell divisions.
I thought it was G0.
Nope, that's interphase. Interphase consists of 3 parts. In G1, the first part of interphase, we've got a new cell. Pretty much it does 2 things during G1. What are those 2 things? It grows and whatever its job is, it does it. Okay? If it's a skin cell, it's keeping things out of your body. If it's a muscle cell, it's moving. It's doing its job. Step 2, S phase. The S stands for synthesis. And during the S phase, what's the main 2 things that happen? DNA gets replicated, and the cell keeps doing whatever it's supposed to be doing, its job, its normal role. And then we get to G2. And G2 is cell still doing its job, but it also replicates what? Organelles. So during interphase, the cell's not dividing, it's doing its job, it's getting ready to divide. It's not part of mitosis. After interphase, this part right here, what's called the M phase, that's cell division. That consists of mitosis, which mitosis is not cell division in itself. Mitosis is division of what? The nuclear material, DNA. And that's always followed by cytokinesis, which splits up your cell. That happens pretty fast. Remember that mitosis takes 1 cell and gives you what? 2 that are identical to each other and identical to the one you started with. It's cloning. Mitosis is like cell cloning. Now, Nolan mentioned a term just a second ago called G0. Remember, in G0, there are some cells that sort of come out of this cell cycle loop, and they just sort of are, meaning that they're doing a job, but they're not ever what? Not ever dividing. They're just there. And we said the best example in your body of cells that do that kind of thing are which cells? Nerve cells. And that's why if you damage your brain, you damage your spinal cord, probably going to be very difficult for it to repair itself. Because one of the main roles of mitosis is repair. If you're not dividing, it's kind of hard to repair. We talked about these terms last time as well. I'm just going to give you a brief definition of these terms just in review. We know a genome are all the genes either in an organism or in a what? A group. What's a group? A population. We talked about the different forms of DNA. Most of the time, your DNA is in this form. It's unwound. It's just sort of snaked out all in your nucleus. That's when it's working. When it becomes this shape, when it's all wound up in what we normally think of as the shape of a chromosome, that's when it's said to be in the form of chromatids. This is a chromatid. This is a chromatid. Together, these 2 together are called what? Bivalent chromosome. What do we know about those 2 sides? They're the same. They're exactly the same. They're copies. Because our chromosomes don't typically become visible to us, even with a microscope, until after what? After replication. So after S phase of interphase. Now, a term that was pretty much new to us before we started this unit was that word right there. We said when we see that term, it looks a lot like the word location. It's the same root. That's what it is. It's a location on a chromosome for a particular gene. If we're ever going to learn how to control genetics and manipulate genes, we've got to be able to locate where a gene's at. We've got to be able to find the gene. And that's what that is. That's a locus. It's a place you would find a chromosome, a locus. These 3 terms we're going to talk a lot about when we get more into meiosis. Somatic cells, we said, are what? Normal body cells. And at least in humans, they're pretty much always haploid or always diploid. Always diploid. And what's the symbol for being diploid? 2N. And our diploid number is 2N equals what? 46. Our germ cells are only found where in your body? Yep. In your ovaries or in your testes. And are they diploid or haploid? They are diploid. But they're the only cells in your body that know how to do what? Meiosis. Meiosis is going to take one diploid germ cell, and the end result of it is going to be what? 4 haploid cells that grow up to be gametes, sex cells, sperm or eggs. So remember that gametes are haploid, N, and in humans, N equals what? 23. It's our haploid number of chromosomes. All right, so that's pretty much all review. And what I want to talk about now is to talk about the stages of mitosis a little bit and how it works. Before I do, though, this picture right here is going to be, again, real important when we get to meiosis, more so than right now. This is a picture of your chromosomes. Now, they've been dyed, they've been colorized, but they're arranged in pairs, homologous pairs. When we talk about homologous, we know that root homo means the same. Are these 2 chromosomes right here exactly the same? No. They're not exactly the same. They're called homologous because they're the same kind of chromosome, right? One of these, though, came from where? Mom. Your mom, and one of them came from your dad, if this was in you. They're not exactly the same, but they have the same what? They have the same genes in the same order, but they may have different what? Different alleles, which are different what? What is an allele? A different version of a particular gene. That word is an important word to know, allele. Alleles are different forms of genes. So homologous chromosomes control the same traits. They have the same genes in the same order, but they may have different versions of those genes because one of each of these chromosomes, one of each pair, came from each of your parents. And another term that we need to know, what's the word for this diagram that we have up here? When we have chromosomes arranged in pairs. We just briefly mentioned this the other day. It's called a karyotype. So remember that root karyo, we've seen that a lot, like eukaryote, prokaryote. That means nucleus, right? It's a picture of the nucleus. It's a picture of the chromosomes from the nucleus, arranged into like pairs, homologous pairs. Another little review here. Here's our bivalent chromosome. Chromatid on this side, chromatid on this side, hooked together in the middle here at this point called the centromere. That's what holds them together. During mitosis, eventually what's going to happen to these chromatids? They're going to split. And as soon as they split and start moving toward opposite sides of the cell, what do we call them? We don't call them chromatids anymore. We call them, say again? Chromosomes, because that's really what they are. Really, a chromosome is one of these, not 2. This is a terminology issue. So here, this slide is just kind of comparing and contrasting mitosis to meiosis. Let's make sure we know the differences. Mitosis, the word actually means dance of the chromosomes. We'll see a video later that's done in time-lapse. Time-lapse photography is where you have a camera that only shoots pictures like every so many minutes, right? And then they run that like a video. So it takes something that happens over a long period of time and it makes it look like it happens in seconds. When you do that with mitosis, it looks like the chromosomes are what? Dancing. They're dancing. They're moving all over the place. But realize that mitosis happens all over your body. It happens from head to toe. Its jobs are pretty straightforward. It makes new somatic cells. And because it does that, it's replacing dead cells. We talked the other day, what are the 2 most fast dividing cells in your body? What are the cells that you have to replace constantly? Skin and red blood cells. You have to make millions of those every day. So they're constantly dying. Also, mitosis helps you to grow. It helps you to repair. And for some organisms, not us, but for some things, it aids in asexual reproduction. Things like amoebas or parameciums or even those lizards that he talked about on the video we watched the other day. What was that called when a multicellular organism can reproduce asexually? Yep, big word, parthenogenesis. If you remember at the very beginning of this class, like the first week or so, I'm running out of space here, I have to curve it a little bit. We read some articles about animals that can do this. Usually they're either reptiles or amphibians.
I don't know.
Yeah, that's the lizard that we saw the other day. Meiosis, on the other hand, is our next topic. Remember that meiosis occurs only in your reproductive organs, gonads, ovaries or testes. It takes a diploid germ cell and eventually splits it into 4 haploid cells. Those haploid cells become your gametes or your reproductive cells. Now, something that it doesn't say up here that we'll stress when we get to meiosis. Are the 4 cells you get after meiosis all the same? No. Go ahead.
So that's what
A gamete and a what? Size, mostly. Polar bodies just die. But another job of meiosis is to mix up the chromosomes, to create what? Variation. And we'll get into how it does that a little bit later. But that's one of the main roles of meiosis, to create some genetic variation. What we want to key in on now are the different stages of mitosis. Juniors, raise your hand if you're a junior. Yeah, I got a bunch of them. She got something she needs to give you. So keep them up so she can see you guys. Thank you.
Thanks.
All right, so let's say a cell is now ready to start dividing. Interphase is ending, G2 is over. It's time to start dividing. The very first stage of mitosis is called prophase. What's pro mean? Yep, you guys put those things up and read those later. What's pro mean? First, beginning, before, sometimes, depending on the root. In prophase, the first stage of mitosis, some things happen to get this ball rolling. Number one, Chad, let's put those up. Number one, the chromosomes, the DNA starts to wind up, and the chromosomes become visible. So the chromatin forms chromosomes. We'll talk about the spindle in a second. We'll talk about what that is. But this network of fibers, looks like a spider web, starts to form inside your cell. And basically those spindles are sort of like ropes, and it's their job to move what around? Move the chromosomes, to split them, to move them, to drag them where they need to go. And then also the nucleolus in the nucleus disappears. And we'll go from there. But that's the beginning of prophase. And we'll see a picture of that in just a second. Step 2 is prometaphase. In prometaphase, the main thing that happens is that the nuclear envelope, the membrane around the nucleus, breaks down. So we're trying to split the nucleus, right? That's what mitosis is. It's hard to break it if it's got an enclosure around it. Another important point is that the chromosomes attach to these little spindles at a point on the chromosome called the kinetochore. It's part of the centromere. It's a part of the centromere that attaches to the spindles. Kinetochore. Also, the spindles connect on the other side to either the centriole in an animal cell or to the cell wall in a plant. And these two things, centrioles and cell walls, act sort of like anchors. Because remember, these spindles are pulling on things. You've got to have a set point, something stiff to pull against. So the anchors are either the cell walls in the plants or the centrioles in an animal cell. So let's look at a picture of what this looks like. This is what a cell looks like in interphase. If you see a nuclear membrane, which we do right here, if you see a nucleolus, which we do right there, and if you don't see chromosomes, right now this DNA is all unwound. We wouldn't even be able to see it under a microscope very well. That's probably an interphase cell. Go a little bit further. Here's a cell that's starting up prophase. You can see the DNA is wound up. We see some chromosomes. The spindles over here are starting to form. And the nucleus is starting to dissolve, the nuclear envelope. Here's prometaphase. Notice the nuclear envelope is gone. The nucleolus is gone. All the chromosomes are now attached to the spindle fibers, and they're attached at a point called the kinetochore. Here's the centriole over here, and here's the centriole over here. These are the anchors for these yellow spindle fibers. The next step is called metaphase. This is the easy one to remember. Meta means what? Middle. Okay? Middle. It's the third stage, but it means middle. And in metaphase, the chromosomes line up in the middle of the cell. They're all dragged to the middle. So in you would have a line of how many chromosomes if mitosis was happening in one of your cells? You'd have a line of 46. We have 46 chromosomes. Right? They lined up single file. And again, they're all connected to the spindles at these kinetochores. What's going to happen next is anaphase. And in anaphase, the bivalent chromosomes get pulled apart. They get ripped apart, and those spindles start to shorten, and they pull the chromosomes toward opposite sides. There's some proteins along those spindles called motor proteins. What motor means what? Moving. Okay? Those motor proteins use some ATP hydrolysis to pull the chromosomes from one side to the other. Your textbook likes to use the word pole or talk about spindle poles. A pole just means what? Well, it means a side. So these chromosomes are getting pulled to opposite sides of the cell. Is all we mean. Before we talk about telophase, let's look at a picture. Here's metaphase. Notice they're lined up in the middle. Here we're only showing 4 chromosomes because it'd be hard to show a picture of 46, right? So we're looking at 4. Centriole, lined up in the middle, all connected at this kinetochore. And then we get anaphase. And in anaphase, those chromosomes, the bivalent chromosomes, get pulled apart. What do we know about that set of chromosomes and this set of chromosomes? They are exactly the same. Exactly the same. This set is getting pulled toward this pole. This set's getting pulled toward that pole. And then lastly, we get telophase. What does tele mean? Like telephone, television. It means far away. Okay? You're getting something from far away. In telophase, the chromosomes reach opposite sides of the cell. They become far away from each other. And after that, it's sort of the opposite of prophase. What happened in prophase gets undone over here. The chromosomes unwind and go back to being what? Chroma 10, unwound DNA. A new nuclear envelope forms around each set of chromosomes. The spindle is taken apart, broken down, and a new nucleolus pops up in each new little nucleus. That's where mitosis ends. Mitosis ends with how many cells? One cell with what? 2 identical nuclei. Mitosis, again, is not cell division. It's division of the DNA, the nucleus. So mitosis ends with telophase, 1 cell, 2 nuclei. Here's what a picture looks like. Notice the cell is 1 cell still hadn't split. It's got a nucleus here, a nucleus here, a nucleolus in each one. The spindles are gone. The DNA is starting to unwind and go back to being chroma 10. What has started already, though, before telophase is over, what's already started here, this furrow? Cytokinesis has already started. It forms this cleavage furrow, this splitting that's starting to happen in the middle. So telophase is very quickly followed by cytokinesis. Remember, cytokinesis is the division of the cell. It happens right after mitosis. And in animals, Think of it like a noose. If this was our cell, there's this group of proteins that goes around the cell in a circle, like a noose. And that circle goes all the way around. And those proteins are contractile proteins, which means they can contract. And they begin to contract, tighten, the noose tightens, and it just tightens and tightens and tightens and tightens and tightens. It's not going to work very well with paper towels, but you get the point. Eventually that tightens up, and the cell's not stiff. It's made out of what mostly? Water and phospholipids. So eventually that tightens up, and this side over here touches this side, and what happens to the cell? Pop apart. So it's pretty easy in animals. It's just tighten, 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 until you eventually just pull it apart. It's a little more complicated in animals, because they've got to build, in plants, right? A little bit more complicated because they've got to build a new what right down the middle. They've got to build a new wall, and in addition, they have to patch what on both sides? They have to patch a new membrane around too. So that takes a little bit more time. It's a little bit more complicated. The new wall, sometimes it's called a cell plate to begin with. That's the beginnings of the new wall. And that's where both the new wall and the new membrane are going to be made. What's called the cell plate in the middle of the cell. But regardless, when mitosis is over, we end up with what? When mitosis and cytokinesis are over, two cells, just like each other, just like the cell we started with. Key point. Can a haploid cell do mitosis? Yes. Yep. Okay. There are some things, remember when we talked about fungi back a long time ago? Fungi are almost always haploid. Do they have to be able to reproduce? Yeah. Okay. If you start with a haploid cell, what do you get after mitosis? Haploid cells. If you start with diploid cells, what do you get after mitosis? Diploid cells. Mitosis makes exact copies. Doesn't matter what you're copying, you get exact copies. Here you can see the cytokinesis happening in the animal cell, how that contractile ring of proteins just continues to tighten and tighten and tighten until eventually that cell is going to get ripped apart. And that's a real picture up there. That's an electron microscope picture. Now here's it's a little bit harder to see, but we're going to have a lab here in a few days. I'm not sure which day it'll be, where we're going to look at under a microscope at cells doing mitosis. And we're going to look at a specific part of a plant. Where in the plant do you think it's growing the fastest now? What part of a plant is growing the fastest? Typically at the tip of one of two places. What two places? I believe tip of the stem or the tip of the root. Okay, those places on the plant are called meristems. It's the tip of either the stem or the tip of the root. We're going to have a slide. We're going to use onions. That's what we're going to be using for our lab, and we're going to use the tips of their roots because they grow fast. We want to use that because if it's growing fast, that also means it's doing what really fast? Mitosis. So we're going to be looking at onion root tip cells, and we're going to be seeing what they look like in each stage. Now this one's it's not a good picture, and it makes it harder even on the projector. This is what an onion cell sort of looks like during prophase, and really all you can see, I know it looks just like chaos in here, but you should be able to see the beginnings of chromosomes forming here. And I know you can't see those very well from where you're at probably. Here, that doesn't look a whole lot different, but you can notice that all of that material is where? It's right in the middle, so that's a metaphase. This one's going to be a little bit easier to see. Here you can see that they've started to split. You've got one set of chromatids going this way, and you got another set moving that way. And then finally, here they've made it to opposite sides. This was one cell to begin with. And what's starting to get a cell plate down the middle, a new cell wall. Here's one new nucleus, and here's the other new nucleus. And eventually this is going to be a hard cell wall, and these will be two completely separate cells, but just alike. It looks very similar in animals. Now, before we get into this regulation stuff, I'm going to take a time out from talking for a minute. And we're going to look at a little Mr. Anderson video clip on similar stuff to what I just talked about. So you'll hear his version of it.
Is that still on the video?
What do you mean?
Watching this, is it still?
Oh, yeah. Now I'm going to take that off. Let me turn this off.